All right, let's get started. So I'm gonna write my name. And first, the question is, describe the structure of a single DNA nucleotide. Well, we know a single DNA nucleotide looks like this. So we know it looks like that. And of course, there's like a OH group here and then an H there, right? So what we're looking at here is the three parts. Describe the structure of a DNA nucleotide. Has three parts. There's three parts. There's one, a phosphate. Two, there's a deoxyribose sugar. And then the third part is there's a nitrogenous base. And then it says, what are the four nitrogenous bases that belong to DNA? Write out the full name. Well, we know DNA uses G, guanine. We know it uses C, cytosine. We know it uses T, thymine. And then we know it uses, I'm like, wait, what? G, C, T, why am I drawing a blank? A, duh, adenine. That'd be embarrassing. So those are the four bases that belong to DNA. And then the next question is, what are the four nitrogenous bases that belong to RNA? All right, so that would be the same, guanine, cytosine. But instead of thymine, RNA uses uracil and then adenine as well. Okay? All right, the next question, what are the two major differences between DNA and RNA? Now, a lot of people say, oh, DNA is double-stranded and RNA is single-stranded, but that's not true necessarily. DNA is typically single-stranded and RNA is typically DNA is typically double-stranded and RNA is typically single-strand. But RNA can fold on itself and can make loops and stuff, and so it ends up, some RNA is double-stranded. So that's not a good designator. But one structural difference we know is that DNA is missing an OH group from its 2' carbon. And we know that RNA uses uracil and DNA uses thymine. That would be the two major structural differences. All right. How many hydrogen bonds are formed between adenine and thymine? Adenine and thymine have 2 hydrogen bonds, while cytosine and guanine have 3. And when we get into Tata boxes, which are made out of adenine and thymine repeats, we'll see why it's important to know that there's only 2 hydrogen bonds there. What is the one rule that DNA and RNA must obey when polymerizing new strands? Well, the polymerase can only put a new nucleotide onto the 3' end of the existing polymer. And so that means that the arrow of synthesis can only go in 5' to 3' direction. So it can only polymerize in a 5' to 3' direction. DNA can be found in a heterochromatin and a euchromatin state. What does each state look like, and when would the cell prefer to have its DNA in one state or the other? Well, heterochromatin is densely wound and tight DNA, and it's used in cell division and when not being used. So it uses it in cell division, but it also, if the cell isn't using the DNA, like if it's a heart cell and it doesn't need the enzymes that a liver cell needs, it'll just pack that away in heterochromatin. All right, the other type is euchromatin, which is loose DNA, so it's not wound around any histones, and it's used in DNA replication. So like if you need to replicate all your chromosomes because you're going to go through mitosis. And then also when RNA is being transcribed. So RNA transcription would be the other time. So you get euchromatin is loose DNA. That's where it's not wound around the histone proteins. And that's used in DNA replication and RNA transcription. All right, what are the only two reasons the hydrogen bonds between complementary DNA strands would be separated? Well, it was this thing above you. It's for DNA replication, right? Because you have to separate the two strands in order to copy them. And then the other one is RNA transcription. So when you need to copy a strand of DNA into a RNA transcript, you have to separate the two strands. So those are the only two reasons that they would ever be separated. On the back side, all right, let's take a look at this. Sorry, this I know it doesn't fill out the full screen, but we'll do our best here. All right, so let's start off at the top. We have direction of replication. That's going to be this big arrow. So let's do the obvious ones first. Direction of replication. So, by the way, this is a figure that shows DNA replication. It's not exactly like the one in the exam, but it's pretty close to it. All right, so we know that the direction of replication is going with that arrow. The enzyme that's unwinding it, we know that's helicase. And then we know the enzyme that's moving ahead of the replication fork is topoisomerase. So we can cross those off. So I cross off topo, cross off helicase, cross off direction of replication. All right, so these two big arrows right here, those are pointing at, these two big arrows, this one and this one, they're just pointing at the two strands. So they're obviously talking about leading and lagging strands. Well, the leading strand is the one where the DNA polymerase does not have to get up. It just polymerizes 5' to 3' without stopping. So that's the leading strand. And then the strand with all the Okazaki fragments, the one where all these enzymes are having to work, because the only way that these enzymes can replicate is 5' to 3' going this way. But the direction of replication, the way helicase is opening, is going this way. And so they have to keep picking up and following helicase along the way. And so that's why this is the lagging strand. Lagging strand. So we got leading strand, lagging strand. Okay, well, we know the enzyme that's working on the leading strand has to be DNA polymerase 3. So DNA polymerase 3, it's going to be that one. We can see that these red fragments here, those have to be RNA primer. And so this little hamburger-shaped pink thing has to be our RNA primase. So I'm going to cross off RNA primase. And then notice this guy right here, he's bound, no, I'm trying to follow my arrow here. He's jumping and binding to the end of the RNA primer and then polymerizing until he gets to the next RNA primer. And he's going to hop up and travel back and so on. This guy right here, this has to be DNA polymerase 3. So that's DNA polymerase 3. All right, this right here, this red patch, that has to be another RNA primer because that's what RNA primase is laying down. So that's RNA primer right here. So that's RNA primer. So we can cross that off, RNA primer. We have DNA polymerase 3. There's a 2 next to it. We've already labeled both DNA polymerases. Now DNA polymerase 1, it's going to be down here. It's going to be down here removing the RNA primer. So that's the DNA polymerase 1. And then here's our DNA ligase. So that's ligating the Okazaki fragments together. And then finally this last arrow here is just pointing at this little fragment. So that's our Okazaki fragment. Okazaki fragment. And that's it. That's everything. All right, last question. How many origins of replication are there in a eukaryotic cell? Many. In a prokaryotic cell, just one. In what state is the chromatin when it gets replicated? It's got to be euchromatin. That's chromatin. That's going back to our original answer back here, where it said, what are the only two reasons that the hydrogen's bonds can be separated? Well, when it's euchromatin, you can do that for DNA replication. So DNA replication is the big one. So in what state is the chromatin when it gets replicated? It's euchromatin. Okay, and that's the worksheet. I hope you guys enjoyed our little walkthrough. Bye.